Well, here we are. It's time for the third installment of our little video tutorials on how the nervous system works and how nerves transmit information from one part of the body to another. And in this one, we're going to be focusing on synapses. Now, it's a Sunday afternoon. It might not be a Sunday afternoon when you're listening to it, but it's a Sunday afternoon now. I'm at home and there are various noises going on around the house, so you'll have to excuse kids, toys, and stuff like that. Our problem is that action potentials can't jump across the gap between neurons. Neurons don't touch each other, so how will these action potentials be transmitted? Well, instead what happens is that neurotransmitters are released and they themselves cross the synaptic cleft. And we're going to look at this process using this acronym, a cav-drab. Say after me, a cav-drab. 1, 2, 3, a cav-drab. That's it. That's our acronym. We're going to look at the stages of nervous transmission across the synapse using these letters. This diagram, I hope, will help us to see how synapses may occur. This is one neuron here, all around here, and it has quite a lot of neurons coming into it. So let's see if we can just highlight a few of this. So this here coming in is a dendrite, and it's coming in and won't quite touch this neuron here. Our action potential will be coming along in this direction, and when it gets here, it would like to get into this neuron here, but it can't because the membranes are not touching, and that action potential can't just jump across. So instead, what we're going to see is from this side of this neuron, neurotransmitters released, they will diffuse across the gap and get to here, and that will kick off an action potential in this membrane. Will go this way, that way, and of course remember this is three-dimensional, so it's going to go over the top this way, over like this. This is not a synapse, but it is a synapse. This is a neuromuscular junction, and it is essentially the same structure as a synapse. It's the junction between a neuron and a muscle cell. Muscle cells conduct action potentials in just the same way that neurons do. This is an electron micrograph of an NMJ, a neuromuscular junction. This, of course, is a transmission electron micrograph rather than a scanning electron micrograph. And you can see various structures within it. MI, those are going to be mitochondria. And the other thing I just want to draw your attention to are the V's here. These are vesicles, and in these vesicles are the neurotransmitter. This is a light micrograph, and you can see a nerve coming in here and then splitting off into various endings here. Indeed, this may be a neuron with several endings on it. A little bit of anatomy of neurons, bit of cytology. I want us to note the difference between an axon and a dendrite. Now, this is a motor neuron. Dendrites bring action potentials towards the cell body. This is our cell body here. The cell body contains the nucleus. That's the nucleus there, people. And then the axon carries it on from the cell body. This here is a sensory neuron, and the receptors is where the action potential is going to start. We're going to get a generator potential in the receptors in the skin, and this will generate an action potential which will shoot along here, and of course this structure here is bringing the action potential towards the cell body, and therefore this structure here is a dendrite. Notice that here the dendrite's actually pretty long and it's myelinated, whereas here the axon going from the cell body to the synaptic endings is shorter in the sensory neuron here. And here in an interneuron, you will probably remember these from GCSE as being relay neurons. The dendrites are positioned much the same way as for the motor neuron, and bring action potentials in towards the cell body here with the nucleus there, and then they go onwards down the axon that way towards synaptic endings, because the relay neuron will synapse with another neuron rather than in the case of the motor neuron, going to a motor end plate or muscle fibers. Motor neurons carry action potentials towards effectors. They may also be glands, like a tear gland. Thank you to biologymad.com. So, let's have a look at that process using a cav drab. We have along here a presynaptic neuron. Now we can see that this is going to be taking action potentials coming in this way. So these are the dendrites. This here is the axon, and it is taking those action potentials down towards another neuron. So therefore, this must be a relay neuron. That's a relay neuron, and this, well, is probably a motor neuron. It could be another relay neuron. Who knows? So, looking at what is going on here in the synaptic knob, let's take our first stage of a cav drab. Number one, action potential arrives at the presynaptic membrane. So we've zoomed in. This is the synaptic knob there, which I've just managed to cross out because I'm a little bit unco with this pen. So here comes the action potential, and it comes along down here. What will happen when it reaches here? Okay, a little bit of background. Ca2+ is held in very low concentration in animal cells. It is pumped out by active transport. So we have ATP going to ADP plus PI inside. And so calcium ions are at a very low concentration inside. There's always low concentration of Ca2+ inside an animal cell, and that is the case for a neuron and certainly at the synaptic knob. When the action potential arrives at the presynaptic membrane, this is the membrane just before the synapse, calcium channels open, Ca2+ ion channels open. You can therefore describe these as voltage gated. They are voltage gated Ca2+ channels. So therefore Ca2+ now enters by facilitated diffusion, because the arrival of the action potential has changed the voltage, changed the potential difference across the presynaptic membrane, and has opened up these calcium channels. So Ca2+ floods in, and that stimulates vesicles containing neurotransmitters to be moved along the cytoskeleton to the presynaptic membrane. Once they get to the presynaptic membrane, then we have exocytosis. They empty their contents into the synaptic cleft by exocytosis. And what is their contents? Well, they contain neurotransmitter. And so we have neurotransmitter put into this synaptic cleft here, which I've represented by NT. Little animation I found. I can't remember where I found it, so I can't accredit it. This is exocytosis going on here. I have no way of pausing this, so you've just got to watch it. We have a vesicle here. It's a phospholipid bilayer. It's a little kind of bubble of phospholipid bilayer, and contained within that phospholipid bilayer, we've got these dots here. That may be whatever it is. In the case of a synapse, this is going to be neurotransmitter, and so neurotransmitter gets emptied into, or emptied outside of the cell, which in this case happens to be the synaptic cleft. Notice we do have docking proteins here on the vesicle. It is protein mediated. Exocytosis and indeed endocytosis are protein mediated and are energetic. They require proteins to do that. If you've seen that wonderful Harvard Cell animation of vesicles being dragged around a cell by little motor proteins, then you can appreciate that obviously this must be a protein mediated process. We've done a CAV. Action potential, calcium channels, vesicles, neurotransmitter, emptying neurotransmitter into the synaptic cleft. So what happens next? Our neurotransmitters diffuse across the synaptic cleft. As soon as they get emptied into the synaptic cleft, there's going to be a very high concentration of them close to the presynaptic membrane. They will now just diffuse down the concentration gradient, if anyone heard the cat in the background there, apologies, towards this side, so that they will be evenly spread out. This is also the reason that the distance between the presynaptic membrane and the postsynaptic membrane is very small. Remember Fick's law, that the rate of diffusion is proportional to surface area times concentration gradient all over, and again I've crossed it out because I'm unco, all over diffusion distance. So if we maintain this distance as small a distance as possible, then diffusion is going to be very, very fast. Which of course is what we want for a synapse. We want nervous transmission to be quick. That's the whole point of it. Once they cross, the neurotransmitters then bind on these receptors here. Note the receptors are only on the postsynaptic membrane, and transmission therefore can only go in one direction. And we're going to get one of two effects on that postsynaptic membrane. And remembering this, we're just thinking about how an action potential can be generated in that postsynaptic membrane. Whether we're going to get, how we're going to change the voltage in that postsynaptic membrane. Let's have a look at the receptors though. They are themselves channel proteins. We've talked at length about voltage-gated channels in the previous two videos, looking at how action potentials are generated and how resting potentials are generated. We haven't really talked about mechanically gated channels. These are the kind of things that you might find in pressure sensors in your skin. Pressure is applied, that forces the channel to be open, and then, for example, Na plus can rush inside like that. In this case, we are thinking about ligand-gated channels, and in our case, we're thinking about it ligand-gated for extracellular ligands. So here's our neurotransmitter. It comes on and it binds to the channel, and as it binds to it, it opens up the gate, and therefore, as this gate is now open, lots of different things can happen. Let me just delete a few things on this here. Once we've opened this channel up, ions will move by facilitated diffusion, depending on what sort of a channel this is and where the concentration gradient is. If this is a K+ channel, we'll have K+, and that will diffuse out. If this is a sodium ion channel, then Na+ will diffuse in. Now you'll recall that if Na+ diffuses into a cell, it will make the charge on the inside of the membrane more positive. And as it makes it more positive, or maybe just less negative, it changes the voltage, which may potentially open up these voltage-gated channels, which are also there. More on that later. Of course, if this is a channel for K+, then K+ will diffuse out like this, and it will make the inside of the cell more negative. And as it makes it more negative, it moves the voltage further away from threshold value. So this is another little animation just of channels opening up. We might have potassium channels which open up, then in K+ out, there we go, carrying positive charge with it as it goes out, or it may be a sodium ion channel which opens up, allowing sodium ions in, carrying positive charges with them. As a consequence, we can either have excitatory synapses, that is, a synapse which makes it more likely that an action potential will fire off in the postsynaptic membrane, or we could have an inhibitory synapse, i.e. one that makes it less likely that an action potential will start off in our postsynaptic membrane. And let's have a look at these graphs to see what it is. Now I'm going to choose to take this line as meaning threshold potential, threshold value. On this graph it appears to be 0. It's not 0. Just ignore that. Just assume this is threshold value here, as discussed in the previous two videos. We're at resting potential here, And let's say we have an EPSP, EPSP being excitatory postsynaptic potential, one of these excitatory neurons. This would be, for example, a ligand-gated Na plus ion channel. As that opens up, Na plus ions flood into the cell, and that raises the voltage, the potential difference across that membrane. Now, if we don't get to threshold value, then that's going to be all that happens, and that's what's going on here. We have an EPSP, doesn't quite reach threshold value, and then we'll just duck back down again to resting potential. Now, let's say then a second EPSP arrives. That opens up more Na plus ion channels, and once we get to threshold value, at this point we get the voltage gated Na plus ion channels opening up, and once they open up we have our all or nothing effect that we talked about on action potentials. So here, if we open up enough channels, we get to threshold potential because enough Na plus has moved in, and then we open up the voltage gated ones, action potential occurs. What's happening with this inhibitory postsynaptic potential? Well, if we open up a load of K plus channels with our neurotransmitters, then our voltage dips down further away, and in fact you're going to get no change from resting potential if you have both an excitatory and an inhibitory happen at the same time. That's what happens if we add EPSPs and IPSPs together. Of course, if we take these to be equally strong, if you have, in this case, 2 EPSPs and 1 IPSP occurring at the same time, you will raise your voltage, but because of the IPSP as well, you won't get a threshold value, and so you won't get an action potential generated. This means that the nervous system can integrate well. You can have one neuron inhibiting a response, whereas another neuron can stimulate a response. And therefore we can have a balance going on and effectively decisions made as to what the response is going to be, depending on various different inputs. It's a great facet of synapses. We're on to B. Hello Andrew, how can I help?
Are you making?
I'm currently recording a video. We might cut this bit out. Okay, cool. Shall I carry on? Okay, we're on number 7, and we're on the B of a Cav drab. Look at that, we're almost there. An enzyme on the postsynaptic membrane breaks down the neurotransmitter. Now this is going to stop the membrane being permanently depolarized. This is going to be very important. And then we reabsorb the products of that breakdown back into the synaptic knob across the presynaptic membrane. And we use ATP to do it. It's going to be active transport, hence we've got loads of mitochondria in the synaptic knob. We're also going to use that ATP to reform the neurotransmitter. Our neurotransmitter that we are looking at is acetylcholine. Indeed, we are looking at a synapse which would be described as cholinergic. Your syllabus, your OCR syllabus, says that you must know about a cholinergic synapse and the events that occur there. And just simply going on, here we have acetylcholine will get broken down to an acetyl group, which you will know as ethanoic acid or ethanoate plus choline. Andrew, are you taking a photograph of this?
No, I'm recording.
You're recording it. I'm being double recorded. There we go. So ethanoic acid plus choline. This is a reversible reaction. This way is the unenergetic way. This is catalyzed by acetylcholine esterase. And this bit here is going to require ATP coming back, and there'll be other enzymes doing that. We come now on to summation, and I'm going to take a little break. Well, on to summation then. Now, it's possible for a neuron to have inputs from a number of different neurons. This is one of the great advantages of having synapses, and therefore the response in this postsynaptic neuron can be determined by the signals coming from these different neurons. And this effect is called summation. Let's look at these synapses here. We've seen these before. Let's have a look at them again. We've got A here, which is an excitatory synapse, B, which is an excitatory synapse, and C, which is an inhibitory synapse. So this is probably opening up K+ channels in D here, these two opening up Na+ channels here. Now we've got these possibilities. We'll have, if an action potential arrives just in A by itself, I'll come back to that in a moment. We're not going to have an action potential starting off in D. We don't get it up to threshold value in that neuron there on the postsynaptic membrane. So sub-threshold stimulation, that's a good phrase for us to remember. Sub-threshold stimulation. Voltage activated sodium channels are not opened. If we just open B by themselves, or rather we get an input just from B by itself, then again we have to say the same thing. Sub-threshold stimulation, voltage activated sodium channels are not opened. A and B together, however, what we have is called spatial summation. Spatial summation means that excitatory neurons which synapse onto the same place have their effects on the same place at the same time, bringing that spatial area, that space, up to threshold value. That is spatial summation, with two or more neurons stimulating the same place. And that's going to lead to our action potential, because we've got enough neurotransmitter there to open up our voltage activated sodium channels because we exceed the threshold value. A plus C, well, no prize of saying that wouldn't get us up to threshold value. Indeed, it results in little change in the membrane potential, and certainly our threshold value is not reached. Now, the interesting thing would be A plus B plus C. So if we had all those three in there, now on this principle, probably that wouldn't fire, because C would be enough to inhibit enough of A and B's stimulation, excitation, to prevent summation occurring enough to start a signal going off in D, start a signal, start an action potential in D. Now there is another type of summation though, and that is called temporal summation. And in temporal summation, let's take neuron A for an example, and let's say that it is only neuron A which is synapsing with D, or B and C, they just aren't doing anything at the moment. Now if we get enough neurotransmitter released from A, or if we get enough action potentials coming down here, and we get enough neurotransmitter released from A sequentially, so action potential, action potential, action potential, action potential coming from A, then we'll get a high concentration of neurotransmitter here in the short space of time. If we get enough neurotransmitter here, we'll open enough channels, and then we will get an action potential starting in the membrane of D. That, when you have a lot of action potentials arriving in a short amount of time, is temporal summation. We're adding things up over a short period of time. Temporal versus spatial summation. Now, back onto temporal summation. The frequency of transmission can be used to determine the strength of signal and various other things. So, summation can also occur due to action potentials arriving at a synapse very frequently along the same neuron. As we've just said, this is temporal summation. And this gives us a higher concentration of neurotransmitter in that synaptic cleft. So we've got higher concentration. That can get us up to our threshold potential, and then we can fire off our voltage gated, well, we will open up our voltage gated channels, get our all or nothing response, as we've talked about. Equally, our frequency of action potentials can also be used to show the strength of a stimulus. So let's say at a higher light intensity, sensory neuron, let's say a rod or cone for the sake of argument, produces more generator potentials. Well, that means we're going to get more frequent action potentials to the synapse, giving more frequent action potentials in the postsynaptic membrane, which will then travel to the brain, meaning that with higher stimulation we get more action potentials coming to the brain, and our brain then interprets this higher frequency as being a more intense stimulus. This might be for light, this might be for sound energy, for any number of things. It might be for pressure. We talked about those pressure sensors earlier on, the mechanically gated ion channels found in pressure sensors called Pacinian corpuscles. So the frequency of transmission can give us an idea of the intensity of stimulus and can also control intensity of response. And we'll get onto that in a moment. Many different neurotransmitters. Now, we've talked about acetylcholine. Acetylcholine is used in most neuromuscular junctions, NMJs, the ones we saw at the start, and also in the parasympathetic nervous system. More about the parasympathetic versus the sympathetic later on in the course. Whereas noradrenaline is the neurotransmitter that is used in the sympathetic nervous system, the one which turns you into Superman. Here is an example of when things go wrong. Clostridium tetani on a transmission electron micrograph. It says TN. Actually, that looks more like an SEM to me. And this here is a patient with tetanus. What's wrong with this poor chap here? He, as you can see from his mouth, he has lockjaw. His jaw muscles are totally contracted and they are not going to stop contracting. He has lockjaw. Likewise, all his other muscles are going. His biceps here, his thighs, etc., etc. Why is this happening? Why is this happening to him? Let's think about that frequency of stimulation question. Strength of contraction in arbitrary units. Now this is a strength of contraction of a muscle, of course. And arriving at these muscles will be action potentials as signified by these arrows here. Time is on this x-axis here, strength of contraction on this y-axis here. We've used arbitrary units because why not use arbitrary units? Now the muscle starts off not contracted, so we'll just, if that's going to happen, it's going to really annoy me. We're going to start off with a low or a zero contraction down here, and as we get one action potential arrive at the muscle, we get a rise in contraction, and then that starts dropping down again, back down to zero in fact, just before the next one arrives, and that comes back down again, and there we go. And so we hop along this way. Now as soon as we get here though, and our next action potential arrives, it goes up, and then another, and then another, and then it's going to start dipping down. Remember, the frequency at which these action potentials can arrive is limited by the refractory period, that time it takes to reset the voltage-gated sodium ion channels. So we have our next stimulation here, back up we go, and up, and up. Well, that's just entirely rubbish. Right, I'm going to rub you out. Eraser, there we go. Let's get this going again, shall we? So up we go, stimulation, stimulation. And let's say maximum contractile strength is here. That's our maximum contractile strength. And we get up to there, and then we get to our maximum, and it carries on the maximum, and then starts dipping down. That's what happens when you have lots of action potentials arriving in quick succession. And that occurs because we get a lot of neurotransmitter in, neurotransmitter in the synaptic cleft and therefore bound to receptors. Okay, but what would happen if we didn't have lots of action potentials arrive, but instead we just left the neurotransmitter in the synaptic cleft? Well, if we just leave it there in the synaptic cleft, we are going to permanently have a high strength of contraction, because that postsynaptic membrane, in this case, would be permanently depolarized. because the neurotransmitter's still bound on to the receptors, isn't coming off, isn't being broken down, isn't being taken back up, we have a permanently depolarized postsynaptic membrane. And that's going to lead to constant maximal strength contraction of our muscles. Now let's go back to our poor chap here. That's what's happened to him, because Clostridium tetani releases a toxin which inhibits acetylcholine esterase. And as acetylcholine esterase is inhibited, so we leave our neurotransmitter, the acetylcholine, in the synaptic cleft, in this case in a neuromuscular junction. It's bound onto receptors, and so we have constant high strength contraction, which is totally disastrous for you if you are trying to live, because you can't open your mouth to drink, to eat. Your intercostal muscles around here are going to suffer, because they're going to mean that you are unable to breathe. They're going to be constantly contracted. A whole load of other reasons. He's not going to be able to move because his legs, his thigh muscles are constantly contracted. So that takes us back onto a cav-trap, our summary. Here it is. A, action potential. Ca, calcium channels. For vesicles. D, for diffusion across the synaptic cleft. R, for receptors. A, for action potential and summation. Just stick that in there. And then finally B, for breakdown and reabsorption of neurotransmitter. Sample question and answer. Here's your sample question. How does the arrival of an action potential cause the release of neurotransmitter at a neuromuscular junction? 4-5 marks. Pause the video, have a go at this, and then look at the 5 bullet points that I'm going to bring up. Pause between these two beeps. Beep. Beep. Here we go with the answers. Number 1, we have depolarization of the presynaptic membrane. Number 2, calcium ion channels open. You could say voltage-gated calcium ion channels open. Number 3, we get this influx of Ca2+ down a concentration gradient. This stimulates vesicles to move to the presynaptic membrane, and the vesicles then fuse with the presynaptic membrane. Well, I hope you enjoyed that video. Thank you.